evening, everyone. We are back again to this class, which I really enjoy. I'm really enjoying teaching this way. And I have my own styles of working and doing things. So in the beginning, gradually we are coming to the momentum. And I hope all of you could understand the questions which I asked, because one day it is going to happen that at the end I might summarize all the questions. Again, today also I am back with some questions. But it's usually, if you see my lecture anywhere else, I usually start with something funny. Meaning I usually have a joke when I start in the beginning. So maybe again I like to start with a joke, and joke again for first year students. And what is this joke telling me is that we had a history professor attending to the freshmen. All the freshmen is first year students. And there were first year students in the class, and the first year students, the teacher was looking at the surnames, that who all are there, and he found one surname just to make the class look fresh maybe, and to start some conversation with the class. He said, "I'm seeing Miss Butcher here. Who is Miss Butcher?" So one student raised the hand. And the teacher asked, "Butcher, you know who is Butcher in England?" She said, "Yes, I know, sir. Butcher is those who were involved in butchering the animals in a long time." And same way he asked, some few other students, like one was Mr. Baker. He said, "Okay, your ancestors were into baking." Then he said, "You are Mr. Smith. Maybe your ancestors were related to some Smith or whatever." Then he saw one very funny sort of surname, and this was it. Can you see this on the screen? Yes. This was this girl whose picture I have picked, and this was her surname. EFXFGAHLTYBGBA. So the teacher was surprised. He asked, "What is this?" He said, "Can you tell me something about your history?" And girl charmingly replied that we made eye charts. Nobody laughed, but I think you should have laughed. You know what is an eye chart? When you go for eye testing, ABCD. Yeah? So that was it. The surname can be anything funny maybe. And there were also, when I was looking for jokes related to genetic code, there are few others which might come in between whenever I am talking about the genetic code. So just to give it a funny start, the teachers they want to generate the interest of the class, and they do a lot of work to get the attention of the class. So I hope all of you, none of you is having as funny surname as this class had. So I come back to my topic today, fourth, that is genetic code. And see, what has happened is that I'm left with fewer classes now. The classes now are fewer, and I have to finish up, and I have to take care that you are going to appear in the exam and you don't miss any important segment or whatever. This is not a part of that segment, but I hope you know what is central dogma of molecular biology. Central dogma of molecular biology makes an important segment of genetic code. What is central dogma of molecular biology? I told you in the previous classes also. What is that? Can you give anyone? There is one transportable mind. What is central dogma of molecular biology?
Translation.
No, that is transcription.
Translation. Yeah. And that process is called central dogma.
Central dogma of molecular biology. Only this process is called as central dogma of molecular biology, or it has something more to this? Any idea? Anybody wants to say? There's something more to this. One thing you should know, that who proposed the central dogma of molecular biology was Krebs. Right? He's a famous man in molecular biology. Right? He's the main man in molecular biology. And another important thing which was associated with the central dogma, which Craig said, but you ignored while paying me this, is that he said the protein is synthesized only with this one-way sequence of reactions. Meaning you will not see protein synthesized any other way in the cell. What does that mean? that for synthesis of protein, it will always be from where? It will start from DNA. That DNA will synthesize the mRNA, will provide a blueprint for synthesis of mRNA, and then mRNA will provide a blueprint for synthesis of proteins. You cannot synthesize protein any other way. That also was an important part of central dogma of molecular biology. It talked about one-way synthesis. Like it included, several possibilities at that time were discussed, like in these three things which are there in this cell. From DNA could be synthesized. Alright, everyone agrees with me here. Then from DNA, mRNA, I am already saying synthesized, but you would have never heard of protein being synthesized from protein or any other route. So that portion also is a part of central dogma of molecular biology, and you must give some focus to this. That and there are several other things here which are gradually, have been discovered with central dogma of molecular biology. But as of now, if I ask the central dogma of molecular biology, the main thing of that is what? What is your name, by the way? Nandini. Very good, Nandini. Well, she's a very good student because I see the enthusiasm in her. So, as Nandini told, it's a one-way traffic, and the other thing is, which I added, is that it has the protein. It is the only way by which protein is synthesized in a cell. Right with this? Then I have another, because I'm talking about genetic code, and I started searching for the jokes for that, and one joke which I found was this one, where God is saying that, of course, I could make man with mud, it was much easier, but I cannot understand what is their genetic code and how can we enact, meaning how can we explain the reasons for that. But humans with several research steps and everything, they could visualize, solve, break this code. Code is what? What is code? Code is the hidden language. If I am in ISI, I hope you know ISI, they will talk in code. I was just watching that movie yesterday. What was that? I tell you what was that. He was talking in codes, India, Pakistan or something like that. So code is what? Hidden language, right? It is a hidden language. So genetic code, how would you define it? It is a hidden language in which the genes are expressed. So here we understand what? The language of genes. Right? You agree, I hope. So if you are asked to define genetic code, is the how the genetic information is encoded. Meaning, what is the secret, After the bioinformatics, what is the main crux is that they try to understand the code, that how the information is stored. So genetic code is again coding or maybe how the genetic information is stored. This is what we study here. Then I checked your syllabus very thoroughly, but I still wanted it to be a very good informative class, a complete class, in a way that even if you talk about genetic code anywhere else, you should not miss anything. So what your syllabus says, salient features mainly. So what I'll do is, I'll first talk about the salient features. Then I have a very interesting question once again for brainstorming, whether you understood genetic code or not. And then I would like to how it was discovered, meaning what was the breakthrough research here, so that we could decipher the genetic code. That I will take briefly, meaning not very much in details. Why? Because we have to go by the syllabus also. We are short of time and we need to finish the syllabus also. So the first characteristic feature of genetic code is that it is triplet. And because I somehow feel that these things are there in the previous classes also, and you are a first year student here, so maybe the universities want to make it gradual. So it's a triplet in nature. The genetic code is hidden and the information is stored as triplet, the word which has 3 words, 3 alphabets. So what we say the first feature of genetic code is what? It is triplet. The genetic code, the ribonucleotide, each base of ribonucleotide, because you would need to write this in the exam. You are expected to write. So ribonucleotide, what happens in the ribonucleotide, each base refers to one alphabet or one information, and it is read as triplets. That we call as that the genetic code is triplet. And these three codes together code for what? One amino acid. And I hope preliminarily you know that in the mRNA, three bases are read at one time, and the transfer RNA comes and finds a suitable amino acid and binds to that and starts the process of translation. That background you already have, right? So we are talking about the features, and the first feature I said is it is triplet. And in the triplet, you know this, that the tRNA will come and carry the amino acid, and it will come and bind in the ribosome, and this is how it happens, right? Then again, coming to the triplet code, when it was discovered, how it was discovered, and when it started in 1950s, it was not that easy or a simple job as you have it now. At that time, people postulated that they knew that we have 4 bases. It's okay that we have 4 bases, but if the code is in the alphabet of 2, then we can explain only 16 amino acids, and how many we have is 20. So we will not be able to explain that. Then 256 becomes too much. It was postulated by Francis Crick at that time that it may be 3 code. It was just a guess, wild guess from his side. Then based on his statistics, and he proposed that it will be 3, and that would be sufficient for 20 amino acids. But it was the genius work of Francis Crick, Barnett and Brenner, and Watts Tobin, who provided the evidence for first solid evidence for triplet code. And how did he do that? Is this this background we don't have maybe. So the triplet code, how did he prove that the code is triplet? Of course, it was proposed on the basis of theoretical speculation. And I have already made a background for you for deletions. You know deletion mutations, point deletion mutations, and you know insertions. You know frame shift mutations, right? And do you know reverse mutations? What are reverse mutations? Reverse mutation. Okay, deletion. Let's start with deletion. Anybody wants to answer what is a deletion mutation? When one base is deleted. What is a substitution mutation? One substitute comes to the another. Right. And what about frame shift mutation? And what about silent mutations? I told you in the last class. Silent mutation. Meaning, of course, the mutation has happened, but it has no effect. Right? So, Francis Crick and his team, they were trying to provide the evidence for triplet code. And how did they do that? They worked with again E. coli, K12 and bacteriophages, and they worked with point mutations, right? They say if I have deletion of one base, and then there should be, if it is one base is there and one right, there should be effect. If 2 are there, then the effect should be. So we basically try to work with the reverse mutations. What is a reverse mutation? Reverse mutation is what? Let's come back to this. Before this, going to the next slide maybe, or we can go to the next. This is the previous one, right? So frame shift, you know what is a frame shift mutation? Is that the reading frame is changed, right? So what did Crick and his team do? He said if it is a one base as a code, then the frame will shift by one base. Meaning one mutation will not alter anything because frame shift के बाद वापस की तो it will be just the same. So he initially, can you see this? Initially in the frame, if we insert one base in the C, can you see this diagram in the television? Is that if we have an insertion of one C, what happens? The reading frame is changed, right? And rest of the bases become out of frame. If the sequence earlier was something, but now it will be absolutely different because one C has come in between, right? And then he said, if at the next step I add another base, then what will happen? The effect of C will be gone. Or if I revert this mutation, it will be gone. And he also said that if there is one deletion, it will be gone because one and one deletion should be gone if it is one base is more important. But if it is 3 base, if it is a triplet codon, what should happen? If 3 bases are removed, then also the effect should go, right? And they did this experiment, and what did they do? When they inserted one C, the effect was there. All frame were disturbed frame shift mutation. It did serve as a reverse mutation. So reverse mutation is what? It can reverse the behavior which had occurred in the previous mutation. What did I say here? I said if you have a point mutation in one RNA sequence, one synthesis, and if you have another point mutation and the effect of this earlier one is gone, that means what? Reverse mutation has occurred, right? So you'll be able to define point mutation, reverse mutation, deletions, right? And Crick and his team said if we can restore the same thing by 3, it means it is a triplet code, right? Because the effect is coming in 3. And this is what is observed. That the frame could be restored back if he inserts 3 bases, right? So this was it, and he could restore, and he proved that we have a triplet basis which serve as the codon. So what was that? The first feature which we studied now is that genetic code is triplet. 3 bases are read at one time. They have the information to carry one amino acid to decide which transfer RNA is going to carry the amino acid. And this was Crick and his team who proved this conclusively with the help of reverse mutation, and they worked with which? They worked with what? They worked with E. coli K12, and they worked with R2 locus and T4 bacteriophage. And one more term which came here was proflavin, which we might have missed, and we talked about this in mutations. What is this? Intercalating. It is intercalating agent. If you remember in mutations, I told you intercalating agents. So what do? They go and insert anywhere in the DNA and they cause point mutation. So Crick and his team studied with intercalating agent proflavin, and they conclusively proved what? That the genetic code is triplet. Right with this, you will be able to write. You will be able to write about this if this comes in the exam. Then explain how will you prove that the genetic code is triplet. You will be able to do that. Right. So the next feature of genetic code is that the genetic code is non-overlapping. Genetic code is non-overlapping, or some people call it as it's comma-less, meaning there is no punctuation is that, meaning 3 are read and then 1 are left and there is a comma, space, nothing. It is comma-less and non-overlapping. What is meant by that? It means, for example, that 1 is read and then it goes to the next. It is not like this. I explain you with this example. Here, if you can see on the board, GTACA, right? If it is non-overlapping, what will happen? GTA will be read, then CA or the next whatever it will be read. And it will never be like this, that GTA is one code, TAC is the other code. This, if it would have been overlapping, then this becomes the code. And if it is the remark or the evidence for this was given was what? If there is a point mutation and if the code is overlapping, what will happen? If this T is ordered in GTCA, then this should affect more than one basis. Why? Because it is overlapping. Sorry, more than one nucleotides, right? Again, let's come back to this again. I said GTACA is this one, and I said it is not overlapping. If it is not overlapping, one amino acid will be decided by GTA and the other one was decided by CA and the next one. But if it is overlapping, what will happen? First amino acid will be divided by GTA and next will be decided by TAC. Alright with this? And then next will be decided by TAC because it is overlapping. Okay with this? Got one point? Again, let's come back to this again. Very nice. So I hope you have this sequence in your mind. GTCA. GTCA is fine with you. I'm giving you this example. I said genetic code is non-overlapping. What do I mean by this? I am saying first three bases are read and then it goes to next three. It is not like this. The first three are read, first is included in first and then the second it starts from the second base. Like the second one becomes TAC. It is not like this. The second one is fourth base for amino acid. And how did, again this was proven? How this was proven was with the help of point mutation. Alright. If there is a point mutation in this GTA, CA, what will happen? By point mutation, if I change T to G maybe, then this sequence becomes what now? GGACA, right? If the code is triplet and non-overlapping, only when amino acid will change, that will be GTA will change. Now, whatever is decided for GGA will come, rest of it will be same. But if it is overlapping, what will happen? GTA will change, or TK will GAC next will also be changed, and third one will also be changed because of one mutation. Isn't it so? And with the help of experiments, it was proven that one point mutation changes only one amino acid. So that was a conclusive proof. With the help of point mutations, it was proven what? That it is non-overlapping. Meaning how it is happening is, first three codons are read and then it goes to the fourth one. Not it starts from the second one. So you will be able to explain the non-overlapping nature of genetic code. Alright with this? And please make diagrams. Maybe I would also have made it here. No, I didn't make. But I just wrote this example and point mutations were used to explain this phenomenon. Is that if the point mutations changes only one amino acid, meaning it's not overlapping. Right with this? And another argument which Crick gave in support of this is that we have limited number of transfer RNAs in any cell, right? We have limited 19, 20. There is a limited number of 18. It varies from organism to organism. I really cannot say it is 18. It varies. The number varies from organism to organism. But it varies, right? But if it is, it gives you much more combinations, and those many transfer RNAs are not present in this cell. So then he convinced that it is non-overlapping, right? The genetic code is non-overlapping, right with this? Then we have next thing is that the coding is again it's a dictionary. It's a proper meaning all the amino acids, what codes for which amino acid gradually was discovered with the help of experiments of Nierenberg and Metal. I hope you have heard about the enzyme peptidal transferase. I will not take that much here because that again goes beyond my scope, which I was teaching you today. Is that he could actually tell which amino acid is coding for, which sequence is coding for which amino acid. Right? So he could actually do this, and so with that one the complete code was deciphered. And we have a proper table where we know the first position in this first position, this is the second position, this is the third position. So you want me to try connecting this again? To see if it connects, otherwise I'll go ahead. So we have a complete dictionary which came out as a result of so the coding dictionary told you what? About each code, please can you maintain silence? Each code has a amino acid for which it is decided. And there are some start codons, there are some stop codons, right? So what are the start codons? We have two start codons, one is UAA and the other one is UAG. And you see that they decide according to the triplet position, and they could conclusively prove by triplet binding assay that the genetic code is triplet, and then they could decide which code is coding for which amino acid, right? And they told us that we have start codons, we have stop codons, and if you can see this here is that one amino acid is being coded by 6 more, 6 like for leucine you see that 6 codes have the same amino acid. So the third feature of genetic code is that the genetic code is degenerate. What does that mean? Is that one amino acid may be specified by 3, 4, 5 codes. It's not that one for one. One amino acid, like I gave you the example just now, is that serine, arginine, leucine, they are encoded by 6 codons. So it varies from person to amino acid to amino acid. And if you see that this degeneracy is apparent from the evident from the pattern also, is that most of the set of the codons specifying the same amino acid are good, such that first 2 letters are the same and only the third letter is different. If you observe that carefully, that the first 2 letters of the codons are same and only the third is different. You won't watch it again. Only first 2 are same and only third is differing in many cases. And for example, the codons for phenylalanine is UU and UUC, right? They just differ in one base, and that usually is the last base, right? That usually is the last base. Like for example, for valine you see 4 codons, and if you see them, they differ only in the last base. For valine, you can see that it differs only for the last base. You got it? It's degenerate. I don't think you have any problem with that. Then came the Vogel hypothesis, and that comes in the exam very often. So what is Vogel hypothesis is that? What is a Vogel hypothesis? Vogel hypothesis is that first and second amino acid are quite decisive. First and second base pair in codon is very decisive, but sometimes the third Vogels, meaning the bonding of the third is weak. Third base is weak very often, and sometimes that is not that accurate. So this hypothesis, Vogel hypothesis, was given by Crick, and he said that the initial 2 ribonucleotide of triplet codes are often more critical than the third member in attracting the correct transfer RNA. Right? The first 2 are more important than the third one. Okay with this? So this is the Vogel hypothesis, and he said that we have 30 different transfer RNA species necessary to accommodate 61 codons this way. Because what is the logic behind this Vogel hypothesis is that cell wants to conserve its energy, and when it is conserving its energy, it requires less of transfer RNA, less number of transfer RNA to perform this job, right? Then next is ordered nature of the code. That the genetic code is ordered. What does that mean? Is that chemically similar amino acids often share 1 or 2 middle bases in the triplet codon. I hope you remember what was silent mutation. Silent mutation is no effect. And why is no effect? Because of the ordered nature of the code. Is why no effect? Because of the ordered nature of the code is that it is the middle one which decides mainly, and the middle one usually is the same in the amino acids which have the same chemistry. Say for example, U or C is present in the second position in the triplets that specify hydrophobic amino acids, right? Including valine and alanine. Or the 2 codons AA and AAG specifies positively charged amino acid, that is lysine. So what is the ordered nature? Basically, cell wants to do what? It conserves the energy. And to replace, usually the chemistry remains the same, and it's the central one, like hydrophilic amino acids such as serine or threonine are specified by triplet codes with C or G in the second position. So the end result is what? Again I said it leads to silent mutations and it buffers the potential effect of the of the triplet code, the mutations on protein functions. Right with this, they are the main reasons for silent mutation. One reason I told you already was reverse mutation. That takes it back. And the another one is the ordered nature of the code. So if anyone asks you what is the main reason for silent mutations or what could be the possible reasons, what would you say? Sometimes it is the ordered nature of the code, and at other times we have reverse mutations. Then we have the next thing is the initiation, termination and suppression. I think that we have given enough time to this one. So we come to the next feature, that is genetic code is nearly universal. Why did I say nearly universal? It is nearly universal. That means it applies equally to viruses, bacteria, archaea and eukaryotes, but not always. It is nearly universal, and we have translation machinery which is very similar in these organisms, and we have cell-free system derived from bacteria, and they could translate the eukaryotic mRNAs, right? So what happens is that in the cell-free system, when it was studied, of course, when I told you during Morgan retired, he was working in the cell-free system, so he could use eukaryotic machinery with the prokaryotic things and interchange. So it is universal, meaning it is same everywhere. But when this work was extended, it was realized that in mitochondrial DNA it is different. In mitochondrial DNA protein synthesis, the genetic code is not universal. It is not valid. For example, maybe I have written one example here. Is that we have UGA, which is termination in the normal cytoplasmic code, genetic code. It is termination, right? But in mitochondria, it codes for tryptophan. So genetic code is nearly universal, but it is not universal. In mitochondrial DNA, it is different. Then we have several other examples. I have several other examples to tell you that it is not universal, and it has been studied methodically and stepwise, and now we have many examples, and I have a paper to show you that maybe UGA is working as termination, but it is tryptophan in each and human mitochondria. We have CUA, which work as UC, but it is threonine in each mitochondria. We have several examples. So genetic code is nearly universal. If anybody asks you, comment on this statement in competitions or anywhere you go. And if anyone asks you that do you agree with this, that the genetic code is universal, what are you going to say? Not always. Not always. For mitochondrial DNA, it is having different code. Then coming to another important concept, which again you will be studying in other classes also, in maybe bioinformatics and everywhere it is going to come, is ORF. What is ORF? The full form for ORF is open reading frames. So what are open reading frames? It is defined as DNA sequence that produces an mRNA that has a start codon and a stop codon. What does that mean? I do it with software very often. Open reading frame becomes what? You are given any mRNA sequence, right? And if you ask your software to find a start codon and find a stop codon and translate this, AUG code for this, UG, and then it will tell you this is a ORF here, right? So ORF is defined by what? Start codon and stop codon and whatever is in between, right? So ORF in general refers to what? One gene, because it is going to synthesize a protein. It has a start codon, it has a stop codon, and we have a sequence in between. That means it is going to synthesize for a polypeptide, right? So ORF is defined as a DNA sequence that produces an mRNA, that mRNA that has a start and a stop codon, which is a series of triplet codons specifying the amino acids making up a polypeptide. In a way, it refers to a gene which is going to code for polypeptide, right? But if you see the figure at the bottom, if you see this figure at the bottom, what does it say? It says a same. See, we talked about the codon being degenerate, meaning commaless. There is nothing in between. But these ORFs is giving you a different concept, meaning that the start codon can lie anywhere. If you see this in this fast FH174, the same RNA sequence is having 7 ORFs. You have this practical in your bioinformatics, is that we give you one sequence and ask you to find out ORFs in this. So it will not be only one ORF, you will find many ORFs there. Alright with this? So we said that the genetic code is overlapping, but a ribonucleotide may have more ORF, more reading frames, and these reading frames could be synthesizing different proteins, different polypeptides. Alright with this? Okay with this? It has an advantage, we discussed that it is degenerate, and if it is not non-overlapping, then one point mutation should affect more than one basis. But one mutation now is affecting different proteins because of different ORF present in the sequence. So these two things are going side by side in a cell and in a system. So this is all which I wanted to tell you, and then I had to skip the Nirenberg experiment because that goes beyond my scope today. But I told you the triplet binding assay, the enzyme peptide transferase, which helped him to decipher the genetic code, and he could work in the cell-free system. Those were the main reasons for genetic code. Okay with this? Right? So we talked about this and we are done. So again, I find this very interesting. Whatever we study, you should have something new in every class when you come for the class. And attending class is also important. Of course, you can view things anywhere. But I always feel that there is a different meaning in one-to-one classes. And right? So brainstorming for whatever we learned today is that I'm giving you one sequence that is wild type. Can you see this on the board? The wild type sequence. Can you see the wild type sequence in the drawing on the right? I'm talking about this. Right? Can you see this? So if first mutant has come, that is, it stopped synthesis at the third amino acid, what has happened? Probably predict the type of mutation that led to each altered protein. Mutant 1, mutant 2, mutant 3. Any guesses what would have happened? That mutant 1 came, only 2 amino acids are brought. What could have happened? Mutation में क्या हो गया होगा? Simple था, सोच नहीं पा रहे आप लोग? It's quite simple. अगर ये wild type था and this is what has happened in first mutate, what would have happened? Stop codon आ गया होगा third position पे. That's a valid guess. Because of point mutation, something would have happened that the third codon is stop codon. Everything, right? So the what would have happened here is you can get when it comes in the objective type sometimes, right? So I said this was wild type, and if we come to what? If we come to the first mutant, what would have happened? The stop codon has come in between and the expression or translation stop. And what would have happened in the mutant 2? देखिए ध्यान से. What if we change? Anyone wants to answer this question? Mike, you go. She wants to say something. My question is, mutant 2 में first observe what changes occur in the mutant 2? There is a change from स्टॉप कोडॉन मुझे इम्पोर्टेंट, लेकिन मैं आपको दिल्ली यूनिवर्सिटी में पढ़ते हैं बड़े ब्रिलियंट स्टूडेंट समझती हूँ और अपनी तरफ से बहुत सिंपल क्वेश्चन लेके आती हूँ, पर I want you to know that you have to understand and apply your things. पहले भी मैंने बताया stop codon would have come, and in the mutant 2 what would have happened? Yes, she's right. Give it back to her. She will explain why did she say why did she say substitution.
मैम सब्स्टिट्यूशन इसलिए क्योंकि म्यूटेंट 1 में टायरोसिन की जगह म्यूटेंट 2 में फिर हिस्टोरिन होंगे.
Perfect answer. So changing one base again would have led to changing one amino acid only. So it is a one mutation, right? Only one amino acid is changed. And what is happening in the third one? Insertion. Where is the change? Where is the change? Can you say this mic? As upper, and what is the result of this? Can we louder, please, bit louder, and explain yourself? So after first there is a mutation in the second code because of only one point change, all the amino acids are changed, and it might be a deletion or addition, but it could be called as what? The frame shift mutation. So the frame is changed, right? I don't know anything. So these were the three things. That was an answer to my first question, where I said predict the type of mutation that led to each altered protein, and you could predict, right? Now the second question is, the wild type RNA consists of 9 triplets, right? You see that there are 9 amino acids which are being coded. Okay with this? What is the role of 9th triplet? One sentence answer. What is the role of 9th triplet here? Yes, please can you give this to me? My diji you बोलेंगे. Only 8 amino acids are here. I said there were 9 triplets and only 8 amino acids. Then what is the answer for this? The 9th triplet was stop codon, right? Agreed? Everybody agreed with this? So that was a very interesting answer to this. And the third question which I have for this is, so this is just a brainstorming. I understand you are not sleeping in the class. You should know how to answer it if the question is asked in directly. Right? And another question which is interesting from my side is, the first 8 wild type triplets, which if any, can you determine specifically from an analysis of the mutant proteins? First 8 wild type triplets, in each case explain why or why not. Can you analyze first 8 wild type of triplets? Why or why? See, you should have, for this one, you should have the dictionary. Which amino acid it was and how it is changed. You'll be able to do it, it's not difficult, right? We have our dictionary to tell you that it requires a dictionary. Alright with this? Then the last question which I found is interesting again, is that it says another mutation, that is mutant 4, which I am wishing to produce, because if I am in a research lab, and I wish to do many things, How do you upgrade yourself in higher classes? Is that a basic foundation is made in your school level. Then we in the college or university, what we do? We prepare you for the research or the higher classes. So we try to bridge the gap slowly. So the fourth question is again trying to check your foundation, and that says what? Another mutation, that is mutant 4, is isolated, and it is amino acid sequences are changed. But the mutant cell produces abnormally low amount of wild type proteins, and specifically, as you can predict, Where is this mutation exist in this gene? You want me to repeat the question? But I want to answer from you. I find you study in Delhi University and I find you. And this again is based on mutation also I did extensively. I have put lot of effort to teach you mutation. You know mutation, now this is genetic code. And what did I say? I said there is another mutant, right? Any mutant. Alright with this? And I said what happened in that mutant is that amino acid sequence is unchanged. Okay? But I said the protein is synthesized in much less amount now. What could be the possible reason or what type of mutation would have occurred in this one? No? Incorrect. Anybody? No reverse mutation. I don't put same mutations. No nonsense. No stop program. Okay? Yes, you are very close to the answer. Mind making error, she is going to answer this. So what could have happened here is? You are very close to this, but the point would have happened is the WLC microbiome, which is now synthesizing much less amount of food. Because the question was that what would have happened in this? Right? I don't have a guide, it is of mind. And all world is a game of mind. It's not listening to anyone. See, the time is gone where you didn't have information, and having information in here was very, very important. But now you should apply your information. So this was just a gist of both the classes, both the units which I have finished is mutations and genetic code. And I try to put my effort, my best that the students should learn and should be able to present themselves if they are given the chance to present themselves here. So again I am done with this genetic code now, and it is going to come in the exam again. I am telling you, and it will be a full-fledged question. Maybe I could say I gave you a few things, but I hope you will be well prepared if you go well in the class and follow whatever is being taught in the class. Anybody wants to ask anything? Any suggestions? Anything anybody wants to say? Anything? Yeah, there is nothing else after this. Thank you. Anybody for any suggestions, any questions, any queries, any problems, any discussions if you want? Yeah, please. Manish.
What will happen is he's having a command.
He's asking something about what we have.
What hypothesis? What is the question is?
What will happen is he's having
Global hypothesis is for all the bases. आप देखिए प्रोडक्ट आपकी फोटो नंबर डाल सकते इसीलिए मैं अपनी मैडम से ये बना के लाई थी क्योंकि मैं आपको चाहती हूँ कि आप ऐसे पूछिए डिस्कस करें ठीक है ये वाली चीजें कोई और नहीं मिलती है क्लासरूम में ही मिलती है आप इन बेसिस को बहुत ध्यान से देखिए फोटो का जो बोर्ड पे है आप ये देखिए ल्यूसीन के 6 बेसिस हैं 6 बेसिस ल्यूसीन को कोड करते हैं तो कहाँ पे फर्क है खाली लास्ट में है देखिए ध्यान से इट इज वर्किंग एवरीवेयर आप इधर देखिए किसी और को देखिए एंड दिस वाज द डिक्शनरी टोल्ड यू व्हाट इट इज यूजुअली द थर्ड बेस व्हिच इज डिफरेंट दिस इज द वोबल हाइपोथेसिस एंड वोबल हाइपोथेसिस नॉट ओनली एक्सप्लेन्स दिस बट समटाइम्स सम changes come and they are not expressed because the change in the third base is not that important because it is basically the first base and second base which is deciding the fate of this polynucleotide. Yes, I like this that he asked this question, and because these question answers make the class live. Of course, I can teach and I've been teaching for 31 years now. Teaching is not my passion, but my passion is to be with the students and come to their level. Meaning, I want to come to your level to teach you whatever way you understand. Right? I heard that a lot of students have gone to their villages because only first year is being taught at this time. So I thought let's not make them miss this thing if they're not here in the class. And I tried my best that everybody is present in the class, they don't miss this time. I understand this is the only time where you enjoy in life, but this is also the only time when you study. This makes the bridge. Now, after BSc only, you will be able to directly go to PhD. And if you see your curricula, you may not be knowing, but I know this genetics paper is not going to come again. It's done. This will not be taught again in BSc. And if you appear for any UPSC exam, if you plan to give medical entrance again, this genetics portion, this is going to be there always. If you give the NET exam, you will see next some questions coming from it. And there are certain portions where you see things are Things are simple, straightforward, but in here things are very tricky. If you see the question was so simple, but you could not answer that. Right? So I think it was a longer class, but I enjoyed teaching you. I hope you do enjoy this. And if you still have any questions, you can ask me. I am your classroom teacher. And maybe you can ask me in the next class also. Thank you very much for being here. And I hope our infrastructure and things get better every day when we are teaching this way. Of course, in the beginning it required more of effort, but I am very hopeful we'll gradually get better. By the time you pass out, you are the guinea pigs basically. You are the guinea pigs for anything. All experiments are being done on you. Next batch will be better and things will certainly improve. So with this, I close this class. Thank you very much for being here. Thank you.